Hi everyone. So today we're going to talk about the metabolism of our proteins or our amino acids. So this will be chapter 18, and we're going to focus on three learning objectives. The first is the metabolic fate of amino acids. So how do we metabolize amino acids? How do they get broken down and turn into energy as a fuel for us? So amino acids have, you know the structure of amino acids, and they have a nitrogen. Well, that nitrogen or the amino group cannot be utilized as a form of fuel. So the nitrogen is excreted in what's called the urea cycle. So our second learning objective is to learn how we take part of the amino acid, the carbon skeleton, and utilize that for energy. And the amino group, how is that converted and put into the urea cycle and excreted as urea because we don't need that nitrogenous waste. And then thirdly, we'll very briefly talk about the pathways of amino acid degradation, meaning how specific amino acids actually enter the citric acid cycle or the urea cycle, how they specifically, all 20 of them, get converted into things that we can utilize as energy or put in the cycle as waste. But first, we want to just review cellular respiration, which is the process by which our cells consume oxygen and produce carbon dioxide. So we have, again, three macromolecules that are carbohydrates, and we know that carbohydrates, or even polysaccharides, are usually converted or broken down into glucose. So we have many molecules of glucose that we get from the food that we eat, either from glycogen that we have stored or from carbohydrates that we eat. And those go through glycolysis, and we end up with 2 molecules of pyruvate for every 1 molecule of glucose. And that happens under anaerobic conditions, but if we want the most energy out of that glucose, then it has to enter the citric acid cycle. So it gets converted into acetyl-CoA by the pyruvate dehydrogenase complex, and then we can get 1 ATP out of that, 1 pyruvate. We can get NADH and FADH2 as our electron carriers, and that goes into the respiratory chain or oxidative phosphorylation. We also talked about last week fatty acids. So we have triglycerols that get broken down into glycerol and free fatty acids. That glycerol enters glycolysis. We know what the fate of glycolysis is, but under aerobic conditions, those free fatty acids cleave subsequently into acetyl-CoAs, and acetyl-CoA enters the citric acid cycle, again giving us an ATP, but more importantly creating NADH and FADH2 that go to oxidative phosphorylation. And so we'll see today that our proteins that we eat get broken down into peptides. Those peptides get broken down into free amino acids, and those free amino acids get converted into things in the citric acid cycle or into acetyl-CoA. And that's how we get energy out of our amino acids, because they get converted into intermediates of the citric acid cycle. And of course, if you're in the citric acid cycle, then you produce FADH2 and NADH, and we'll see how that actually enters the respiratory chain. And so for our amino acids group, we're going to see quickly how they enter the citric acid cycle, stage 2 of getting energy out of those proteins, before we talk about stage 3 in the next lecture. First, let's set up a few facts about amino acids, or utilizing amino acids as energy. Not all organisms use carbohydrates and fats as a source of energy. Our body is adapted to use carbohydrates very well as a source of energy, and we also can use fats that we have stored as a source of energy. But our carnivores, such as our lions and tigers and things like that, they get about 90% of their energy needs from protein. They get some carbohydrates, from maybe the glycogen if they eat the liver of the animal that they eat, but most of their energy needs are met by eating protein. And so we are looking at how those and how we actually convert proteins into amino acids and convert amino acids into energy if that's what we need. Only a small fraction of herbivores, such as this zebra here, needs are met by amino acids. So they're mostly eating grass, which is a carbohydrate, or some other vegetation, which is a carbohydrate. And so they really don't get their energy needs met by proteins. Microorganisms that maybe live in our soil and our environment can scavenge amino acids from their environment for fuel, but they don't utilize any more amino acids than they actually need. And what they need, they usually convert those into enzymes that they would use to metabolize or do all the things that they need to function. Since our body's main choice of fuel is carbohydrates and maybe fats, we only utilize amino acids for catabolism under 3 circumstances. And the first is where there are leftover amino acids from normal protein turnover. So first, we can utilize free amino acids, of course, to build up our peptides and our proteins. But if there are amino acids left over from normal protein turnover, that's how we utilize amino acids to make up peptides, to make up proteins. We can utilize those under oxidative catabolism or for fuel. We can also utilize amino acids when the amount of dietary amino acids exceeds the body's protein synthesis needs. And so many of us may eat more protein than we actually need to synthesize the enzymes and proteins for our body. And so we can utilize those proteins for fuel only when we need that for fuel, or when there's a carbohydrate or fat shortage. So first, our body really likes and knows how to utilize carbohydrates and fats for fuel. But say you're dieting and you have a high protein diet and you have a shortage of carbohydrates, then your body can easily go into using those proteins for fuel. Amino acid oxidation provides energy through the citric acid cycle, sometimes called the TCA cycle, or oxidative phosphorylation, but it does cost energy to do that because we have to eliminate the nitrogen. Only the carbon skeleton can be utilized for energy. So our body is adapted to not want to first utilize proteins for fuel because it costs energy, and our body wants to save as much energy as it possibly can. So only under these 1, 2, 3 circumstances would our body actually turn to proteins for fuel. And our body does prioritize the use of proteins or amino acids. The first priority when we ingest protein is to synthesize and recycle. So protein turnover means that our proteins are constantly being synthesized, broken down, and resynthesized. So we can have proteins that have a certain sequence to make up our enzymes and make up our proteins, and then they can be broken down to free amino acids and then built back up in another sequence to make another enzyme. And so that's called protein turnover. And so when you eat protein, your body's first utilization of that protein is to say, oh, I need more enzymes, I need more protein, I need more whatever. And we'll break those down into amino acids and build those back up into our protein needs. The second priority for amino acid is catabolism. So once our body is sure it has all the amino acids it needs to function properly and make the proteins and enzyme that it needs, then it will use those protein in a diet for catabolism or for energy. If you do have a diet that's rich in protein, like a keto diet or some other kind of diet, or you just eat a lot of meat, our diet rich in protein leads to extra amino acids, more than we actually need for our protein needs. And so we would actually remove the amino group because that is not used as a fuel. Take the carbon skeleton, and the carbon skeleton is called an alpha keto acid, and we'll look at that very shortly. And then that alpha keto acid is pushed into the citric acid cycle, similar to how carbohydrates and fatty acids are pushed into the citric acid cycle. So if you have a diet that's high in protein, you would remove the amino group. The amino group gets excreted as urine in the urea cycle, and the carbon skeleton enters usually the citric acid cycle. These are the only really two ways we use our proteins. We never ever store our proteins. You never store proteins. You eat too much protein. What happens is you remove the amino acid. You might enter the citric acid cycle, or you might go through the process of creating acetyl-CoA so that you can go back and produce fats for storage or glucose for storage. Because we can't store protein, we have to remove the amino group and store it as the carbon skeleton in the form of fats or carbohydrates. So let's start from the top. What happens when you eat meat or some other protein? Well, we know that protein goes through our throat, it goes down our esophagus, it goes into our stomach. And then tucked behind our stomach, we have this little organ called our pancreas. And our pancreas is going to be activated and it's going to start secreting proteases. And those proteases are going to start digesting the protein. So the pancreas is going to start through a pancreatic duct, is going to put proteases into our stomach so we can turn those into peptides, so we can turn those into free amino acids, so that we can turn those into energy or some other form that we can store our protein as. And so our proteins actually exist in two forms. And so the two forms of our proteases of course are enzymes that break down proteins. They exist in two forms called zymogens. So a zymogen just means that our proteases, when we're not trying to digest a protein, it has one inactive form, and then usually by the process of phosphorylation, those proteases get into the active form where they actually can digest our food, because we don't want always our protease digesting things in our stomach and our pancreas. Actually, if you start prematurely digesting, have active digestive enzymes, they would start digesting our actual organs like our pancreas. So our body has to have a way to have inactive and active forms of proteases so we won't actually digest our organs. Like I last year had pancreatitis, which means my pancreas was producing too many enzymes like lipases and amylases and proteases, and they start to digest your pancreas and damage it. And so we have to turn that off so we won't cause damage to our pancreas. But anyway, our zymogens mean that we have inactive and active forms of our proteases, and we utilize the prefix pro or the suffix ogen on a protein, means it's in the inactive form, and then when they get turned on, usually by phosphorylation, they become active. And so what happens when that protein starts to go down into your stomach? Well, first in our stomach, our body recognizes that we need to digest a protein. So originally the pepsinogen that's in our stomach gets activated and turns into pepsin. So this is an active protease, and we've eaten some meat or some beans, and so that pepsin that's now activated at a low pH starts to break down that protein into peptides. So we know that protein, short forms of protein, is peptides. Alright, so then we have other proteins. When they start to pass into the small intestine, those peptides pass through the small intestine, the pH is different, they get activated, so trypsinogen or chymotrypsinogen gets activated, and we get trypsin and chymotrypsin. So you don't necessarily need to know the names of these different proteases, but it's good just for your repertoire of knowledge just to be familiar with the fact that we have different, very well known or very well studied proteases that get activated, and they go into their active forms and cut our proteins into peptides. And then more proteases come and cut our proteins more specifically into smaller and smaller peptides. And finally, we would have aminopeptidases or carboxypeptidases that get activated. And of course, our aminopeptidases would start cleaving those smaller peptides from the N-terminus, and our carboxypeptidases would start cleaving those peptides from the carboxy end. And then we would have free amino acids. So again, we start in our stomach, we start breaking down those proteins into peptides. More proteases break those peptides into smaller peptides, and then aminopeptidases and carboxypeptidases break those peptides down into free amino acids. And so the net result is that we will have free amino acids that are absorbed into the blood through the intestine. So now that we have free amino acids, they can freely go through the bloodstream and go wherever they're needed, so they can be turned into energy, or so they can be recycled into new amino acids for protein turnover. So now that we have free amino acids, they can actually go into two cyclic pathways. We saw that citric acid cycle was a cycle, but the urea cycle, where we place our nitrogen, is also a cycle. And so we'll look at that. So we can have intracellular protein that need to be broken down and built back up, or we have dietary protein, and we just saw that the dietary protein get broken down into peptides, and then they become amino acid. And so what we said is those amino acids have two parts. One is the carbon skeleton. That's the only one that we can actually use as energy, or we can actually store. So that carbon skeleton gets converted into alpha keto acids, and that can go into the citric acid cycle, where we know we can get energy out of it. Also, we just need to store that. Then we can convert it into oxaloacetate, which can go into gluconeogenesis cycle, which can give us glucose, and glucose can be stored as glycogen. So again, we never store our amino acids, but what we do is take that amino acid, break it down into two parts. One is a carbon skeleton, which is an alpha keto acid, which can enter the citric acid cycle and give us glucose and then glycogen. But what do we do with that amino group? Well, we cleave it off as ammonia. That ammonia can be utilized to synthesize other amino acids. That ammonia can be utilized to synthesize, we'll talk about later, carbon 1 phosphate, and that enters the urea cycle, and then it gets excreted as urea. And so we have, our amino acids have a fate, but it has two fates because it's broken down into ammonia and its carbon skeleton. And the ammonia, or the amino group, is excreted from the body. So our nitrogen removal from an amino acid is an amino transfer. So we're going to do this amino transfer, and we're going to learn it in just one step. So let's start down here, because I think this figure is a little bit confusing in the way it's drawn. We have our free amino acid. So whatever the amino acid is, there are 20 of them. There's glycine and alanine and tyrosine, whatever. They have different R groups, and they all have an amino group. We need to remove this amino group because this amino group is waste. It's nitrogenous waste, so we need to get rid of it. How do we get rid of it? We actually take it and transfer it to alpha ketoglutarate. Where does alpha ketoglutarate come from? We've seen alpha ketoglutarate in the citric acid cycle. So in your cells, you would have alpha-ketoglutarate because we produce it as an intermediate of the citric acid cycle. It comes from other sources as well, but we know it as an intermediate from the citric acid cycle. So again, we need to get rid of this amino group off of our amino acid. We just transfer it to an alpha-ketoglutarate. What enzyme catalyzes this reaction? An amino transferase. So this amino transferase can be specific or inspecific. It's just usually specific for a particular amino acid, like an alanine amino transferase would catalyze the reaction of transferring an amino group from alanine to alpha-ketoglutarate. The other thing you should note about this is that it has a prosthetic group, and I'll just write that out. PLP, you don't need to know the name of it, but PLP is a prosthetic group. I think I'm spelling this right. All right, so that PLP there, it just means that the transfer is happening from this prosthetic group that's attached to the amino transferase. So the PLP grabs the amino group, it throws it on to an alpha-ketoglutarate. And what do we get when we add an amino group to alpha-ketoglutarate? A glutamate. So we have transferred our amino group to alpha-ketoglutarate and made a glutamate. And what's left of the amino acid is the alpha-keto acid. Is the alpha-keto acid here. And this part we can use because we can take this alpha-keto acid and we can put it into, I'll just call it the TCA cycle or the citric acid cycle. So we have one part of that amino acid we can use, and the other part, the alpha-keto acid, we can use because we can put it into the citric acid cycle, and we can get energy out of that there. But the amino group we can't use, so we add it on to an alpha-ketoglutarate with the help of amino transferase, which has a prosthetic group called a PLP, and we make a glutamate. So why in our bodies, like our muscle cell, do we go through the trouble of just adding this NH3 to the alpha-ketoglutarate, and making a glutamate? Well, it's because, let's say you broke down some muscle cell for energy and you had to remove this amino group. Well, what's left? It's usually going to be an NH4+, which is ammonia. So you don't want ammonia going through the bloodstream, because it's going to go eventually, we'll see, to the liver so it can be excreted. You don't want free ammonia going through your body, because ammonia, just like all disinfectants or cleaning products, will kill bacteria and viruses and stuff like that, but they will also kill you. So ammonia is toxic, and so we don't want it going through our bloodstream. And so our body goes through a lot of trouble to take this this amino group, and attach it to an alpha-ketoglutarate so we can make a glutamate. And so then we can safely take this ammonia, which is toxic, it will kill you, put it onto something that can safely go through our bloodstream so it can make it to our liver and then be metabolized or excreted there. Okay, so now we've created this glutamate. Remember, we took our free amino acid, we had to take off that amino group because it's waste, and we safely want to carry it to the liver. So that's where the process is going to happen so that we can actually excrete this amino group, right? So again, we don't want to carry ammonia through our bloodstream if this is happening in the muscle cells or whatever, because ammonia will kill you. Well, we want to safely carry it to the liver so we can excrete it. And the liver has all the enzymes so that it can do that. And so we have this glutamate, And then we're going to perform an oxidative deamination, and that occurs in the mitochondrial matrix of the liver cells. All right, so if we have an oxidative deamination, we remember that oxidation is loss. All right, so I'm going to lose some electrons. Who am I going to give those electrons to? Well, I'm going to give them to an electron carrier, and that's either going to be FAD or NAD plus. So I'm going to actually give those electrons to NADP plus or NAD plus, either of those, whichever one I have most abundantly around in my liver. So I'm going to give those electrons to NAD plus, and I'm going to create this now oxidized intermediate. And then I am going to deaminate. So I am going to take off this ammonia now, and I'm going to replace it with an oxygen. So we have this, and then recreate alpha ketoglutarate. So I would say don't worry about the intermediate, but know that my glutamate dehydrogenase, hope I'm not skipping all over the place. Know that I'm going to utilize a glutamate dehydrogenase, so that's simple to remember because I'm doing an oxidative deamination. So I need a dehydrogenase to do oxidation or reduction. So I've cleared this glutamate all the way through my body. I got to the liver. I need to do my oxidative deamination. Who's going to help me do that? My glutamate dehydrogenase, because it's an oxidation. If I'm oxidizing, I'm going to lose electrons. Who am I going to give those electrons to? I'm going to give them to NADP plus or NAD plus, either one. And then I'm going to get NADPH or NADH. I'm going to create this intermediate because I had an oxidation reaction. But then I'm going to do my deamination. So I'm going to deaminate, lose my amino group. It's going to leave as ammonia in the liver, and the liver is going to know what to do with that. And once I've done that, I'm going to go from glutamate to alpha ketoglutarate, which I can use now in my citric acid cycle again for energy. So we know what to do with our alpha ketoglutarate, and then we're going to see what to do with this ammonia group, transfer it into the urea cycle so that we can get it out of our body as urea in our urine. We just saw how we take most of our amino acids and take off the amino group and add it onto alpha ketoglutarate so we can have glutamate. Glutamate can remove the ammonia then in the liver. But sometimes if we have vigorously working muscles, we're working out or something like that, they operate almost anaerobically, so there's not enough oxygen around to actually convert the glucose that it breaks down for energy into pyruvate and change that into acetyl-CoA that can go into the citric acid cycle. And the citric acid cycle needs oxygen. And so if we have vigorously working muscles, this is a arm muscle, and we're going through glycolysis because we're breaking down the glucose and the glycogen, and we're creating pyruvate in the muscle cells. Well, that pyruvate, if it builds up too much under these anaerobic conditions, they will make lactate and then lactic acid, which can cause problems. People know about the problems caused by the buildup of lactic acid, so we don't want lactic acid built up in the cells. And so what can happen is that pyruvate can be converted into alanine. So here is the structure of pyruvate. Hopefully you remember the structure of pyruvate. And here is the structure of alanine. And so what happens is you have pyruvate, you don't want it to turn into lactate because that builds up lactic acid. So what you do is you would transfer the amino group from glutamate in your muscle cells utilizing an alanine amino transferase. So you take the amino group off the glutamate, you add it to the pyruvate, eliminate the water, and then you get alanine. And so this is called the Glucosealanine cycle It's from a buildup of pyruvate in vigorously working muscles that alanine can safely carry that amino group from glutamate through the bloodstream and take it into the liver. And so then in the liver, that alanine will transfer that amino group again to glutamate, creating pyruvate. And we can take that pyruvate and create glucose, and we can keep utilizing the glucose for energy. So the alanine, once it's made it to the liver, safely transporting that amino group, utilizes an alanine aminotransferase, takes that amino group, adds it to glutamate. Glutamate can then safely go and convert it into ammonia, which goes through the urea cycle, and then pyruvate can be converted then into glucose through gluconeogenesis. So this is all happening under anaerobic conditions where we're safely converting alanine into pyruvate into glucose, which can give us energy under anaerobic conditions. Your alanine aminotransferase is a very well-known aminotransferase, and it, on medical terms, if you are having some issues and they want to check your liver function, they will check for alanine aminotransferase levels, and if they're too high or too low, that means your liver is not operating as it should. So that's like a medical thing that we use for our alanine aminotransfers or aminotransferases in general. If you go to the doctor right now, I just recently went to the doctor and they checked my alanine aminotransferase levels to see how my liver was functioning. And so this test for liver function, and why? Because you need alanine aminotransferase to take off that amino group and give you pyruvate so that you can create glucose so you can continue to work under anaerobic conditions. So we just learned that everything, or usually everything, leads to glutamate. Glutamate is the transporter of that amino group from all our amino acids. Where does it transport it to? It transports it to the liver. And in the liver, we know that we can utilize our glutamate dehydrogenase to get rid of that amino group as ammonia, and that ammonium goes into the citric acid cycle. So here we are, and we are in the liver. So now we're in the liver. Already in whatever cells there were, this free amino acid took off this amino group, circled the whole thing, took off this amino group and added it onto alpha-ketoglutarate, and we have glutamate. If we have too much pyruvate building up in the muscle cells, then we created alanine from pyruvate, and then once we get to the liver, this amino group adds on to alpha-ketoglutarate, and then we have glutamate. So now we're in the liver, in the cytosol of the liver, and we have a bunch of glutamate molecules. We also have some alpha-ketoglutarate and some alpha-keto acids. Those can go into the citric acid cycle. That's fine. But now we have a buildup of glutamate. So what do we do with that glutamate? Well, that glutamate has to pass into the mitochondrial matrix, because that's where the enzymes to process that ammonia are. And so we also learned that the glutamate takes off that ammonium utilizing a glutamate dehydrogenase. It's an oxidative deamination. So you have your oxidative deamination in the mitochondrial matrix of the liver cells or the hepatocytes, and you have your ammonium here. So then we're going to learn what happens to that ammonium. There are other amino acids that are processed a little bit differently. If we look at glutamine, we know that glutamine, our amino acid, has two amino groups on them. And so if we take off one of those amino groups, catalyzed with the enzyme glutaminase, then we take off one, we get glutamate, and we know that glutamate utilizes glutamate dehydrogenase and creates ammonia. Now we have a free ammonium, and that is processed just like it would be for the free ammonia we create with glutamate dehydrogenase. We also might have some oxaloacetate that's in the mitochondria of the liver, and that oxaloacetate can take the amino group and create aspartate if we need that. But we're not going to worry about that right now. Now we're just concerned with, we have all this glutamate, we utilize glutamate dehydrogenase, we created ammonia, or maybe we created ammonia from the extra amino group that was in glutamine. What do we do with it? And so that's where our carbamyl phosphate is created. We'll go over this in a little bit more detail in the next slide. But we're utilizing 2 ATP molecules. So it costs energy to actually process this ammonium here. We have to take the ammonium, we add in this bicarbonate molecule, and we utilize the 1 phosphate from an ATP, and we create a carbamyl phosphate molecule. And so you should know the structure of this carbamyl phosphate molecule. It utilizes basically a CO2 from bicarbonate, a phosphate from ATP, and the NH2 from the ammonium that we have left over from the oxidative deamination of glutamate. So what we just did is create carbamyl phosphate. So the carbamyl phosphate here, it takes that ammonia group that we got from glutamate. So we're going to say we had an ammonium from glutamate, and we need to get rid of that. And then we added it to a bicarbonate. So our carbon from our bicarbonate here plus HCO3. And then to it also a phosphate. So that phosphate comes from 2 ATPs. And the mechanism of that you don't need to know, but just know that we need energy to actually get rid of this ammonia, and that's going to come in this step. We're adding 2 ATPs. One of the ATP is going to donate a phosphate group to this carbamyl phosphate, and the other ATP is just for energy. So we created a carbamyl phosphate from a synthetase, carbamyl synthetase, which you don't need to know the name of. All right, so now we are carrying this ammonia in the form of carbamyl phosphate, and we had to have a lot of other things in there to help us to do that. So now that we're in the mitochondrial matrix of our liver, we are going to carry that ammonia through the urea cycle so that we can excrete it as urea. And so normally you have to learn the enzyme and the structures and all that. It's late in the semester, so we're going to skip learning the structures and just kind of learn the steps of the urea cycle because these structures are really complex and unnecessary. All right, so what do? The first step, when we have our carbamyl phosphate, we want to get it into the urea cycle. And so we're going to utilize an enzyme called ornithine transcarbamylase. So this is the enzyme that's catalyzing the reaction. And so reaction 1 is basically going to just be a condensation. We're going to take our carbamyl phosphate, I'll just see CP, our carbamyl phosphate is carrying that ammonia for us, and we're going to basically smush it together or condense it with an ornithine. All right, so it's going to condition with an ornithine. And what is ornithine? If you look at it, it is actually just an amino acid. So this amino acid is not one of our standard 20 amino acids, but it's just an amino acid, and we get it from somewhere, and it ends up in our liver somehow. So we have this ornithine, and we add it to our carbonyl phosphate. We know how we got that, and the enzyme that's going to catalyze this reaction is called ornithine transcarbamylase. This is not a standard abbreviation, so you shouldn't use this as a standard abbreviation. The name of the enzyme is called ornithine transcarbamylase, and you get citrulline. So you don't need to know the structure of this citrulline. Just know that basically when you smush together my ornithine with my transcarbamylase, when you smush together your ornithine with your carbonyl phosphate, you get citrulline. And we notice that the citrulline does not have phosphate in it. So we're going to lose our phosphate. All right, so we had a lot of trouble putting it on there. We had to use 2 ATPs, but we needed the energy of breaking that bond to do that. All right, so now we've created citrulline. The next step, step 2, I'm going to start with the enzyme first here. The enzyme is called arginosuccinate synthase. Okay, so arginosuccinate synthetase, and it does what it suggests it does. It's going to synthesize an arginosuccinate. So in the end, we're going to get a molecule called arginosuccinate. And so you don't need to know the structure of it. Just know that I'm going to take a citrulline and I'm going to do some stuff with my arginosuccinate synthetase, and I'm going to get an arginosuccinate. All right, so what to notice about this arginosuccinate is part of it comes from aspartate. So if I take put this here, let me use another color. I take my citrulline, and I'm going to add to it an aspartate. And I'm going to get this arginosuccinate utilizing the enzyme arginosuccinate synthetase. All right, so this aspartate comes from somewhere and it gets into our liver, and we use it, and we combine our citrulline with our aspartate and get our arginosuccinate. Now you see some other things happening here. We have to utilize the energy of ATP to make this reaction happen. It doesn't matter what's happening. Just know that we're going to have to input ATP on the reaction side. So a mechanism that's complicated and necessary. We're going to use up some ATP again and get an arginosuccinate, something that looks like a succinate stuck to an arginine. When actually we're not utilizing an arginine as our substrate, utilizing an aspartate. So that can be kind of confusing. Okay, so then our third step, hopefully I can get it all on here. Our third step is going to utilize an arginosuccinase. So for our third reaction, so let's go to reaction number 3. Use this color. We're going to utilize the enzyme arginosuccinase. Okay, so we're starting with our, just going to call it AGS. We're starting with our arginosuccinate. We got it from the condensation of our citrulline and our aspartate that came from somewhere. And this enzyme is going to catalyze reaction where we have fumarate leaving and we're forming an arginine. And you know the structure of an arginine. So in the end, what's going to stay in the cycle is going to be an arginine. again catalyzed by enzyme number 3, or arginose-succinase. But what's going to come out is our fumarate. So we see this very large arginose-succinate, and we say what stays in the cycle is the arginine, but what leaves the cycle is a fumarate. So what happens to that fumarate? Well, fumarate can go into the citric acid cycle, so we can use that and put it somewhere else. We don't need it, we get rid of it. It's not in the urea cycle. What stays in the urea cycle is that we have an arginose-succinate, and arginine goes on to the urea cycle. All right, so now we got this arginine, which we know the structure of, we can deal with, but it still has these two ammonias on it. We said we wanted to get rid of that ammonia. So how are we going to get rid of that ammonia? We are going to create a urea. And so we started with our arginine. All right, we're going to utilize our arginase, and we're going to lose a urea. Urea is a very simple molecule. It's in your urine. It makes it yellow. That's what it's for. We get rid of that. We can excrete that out of the body, and what's left in the cycle is our ornithine. And we said it's a cycle, so our ornithine gets regenerated, so we start with the same thing that we end with the same thing that we started with. And then ornithine can go condense with an carbonyl phosphate, create a citrulline, and it can go on through the cycle. So those ammonia, our ornithine has an amino group on it, but we got rid of 2 of our amino groups in the form of urea, which can be excreted from the body, and we're left with ornithine, which we know we should end up with because it's a cycle and we need to regenerate that. All right, so again, we're going to remember our enzyme names. We're going to remember our substrates and our products and our leaving substrates, but we're not going to worry about the structures of these things. So let's just summarize what we did. We created a carbonyl phosphate using a synthetase. We had to put in our ammonia that came off our glutamate. We had to use a bicarbonate. That's a bunch of that sitting around our body. And we had to utilize 2 ATPs to put a phosphate on there. When we condensed it with ornithine, utilizing our ornithine transcarbamylase, we got citrulline. Our citrulline combined with aspartate. We had to use an ATP to do that, but citrulline combined with an aspartate, utilizing an arginose-succinate synthetase, and surprise, we get arginose-succinate. All right, so then our arginose-succinate basically breaks down into 2 things. Arginose-succinate breaks down into 2 things. One is fumarate, which goes on. This fumarate goes on, to the citric acid cycle, and the arginine stays in the urea cycle. So now we're at our last step. It's only 4 steps. Arginase catalyzes the reaction where our arginine breaks down into urea, which can be excreted from your body, and ornithine, which stays in the urea cycle and continues on and on. So now we've gone through the urea cycle. That was this objective 2. Last objective, which I'm going to do very, very quickly, is how we get amino acids, specific amino acids, into the citric acid cycle. So don't worry about memorizing this too much. Just know that there are a lot of places we actually get catabolism of our amino acids. And so here we have many of our amino acids, which arginine, glutamine, histidine, proline, they get converted into glutamates, and the alpha ketoglutarate form goes into the citric acid cycle, and then the amino gets deposited into the urea cycle. Some of our amino acids can enter as succinyl-CoA. A couple of them enter as fumarate. Many of them eventually enter the citric acid cycle as oxaloacetate, and others still enter as citrate. And so we see there are lots of different fates of our amino acids. Some of them are glucogenic, meaning they enter into the citric acid cycle and get metabolized that way. And some of them are ketogenic, meaning they can enter into the citric acid cycle or form ketone bodies. And if we remember, our ketone bodies are acetone and acetoacetate, and the other one, which can be utilized in our brain under starvation conditions when there's not a lot of glucose around. But this is not something you should necessarily learn right now. It's more to tell you that we have 20 different amino acids, and they have lots of fates, mostly entering into the citric acid cycle at different steps or as different intermediates in the citric acid cycle. So this is what you actually should learn. This is a summary of the previous slide, the intermediates of the central metabolic pathway, how our amino acids are degraded and catabolized. And so some of them are ketogenic amino acids. They convert into ketone bodies, as I said, and some of them are glycogenic and can be converted into glucose. We know that if we can get something that's oxaloacetate, then they can be turned into glucose in gluconeogenesis. So there are 7 of them that can be converted into acetyl-CoA. There are 6 that can be converted into pyruvate. There are 5 that can enter as alpha-ketoglutarate. There are 4 here that can enter as succinyl-CoA, 2 as fumarate, and 2 oxaloacetate. So if we have our 20 amino acids, then they can enter the metabolic pathways at different steps in the citric acid cycle. And so you should focus on the summary of how they can actually enter the citric acid cycle. So here's a summary of what we learned in chapter 18. I won't read it to you, I'll leave that to you. But we learned how we break down our proteins into peptides, into free amino acids, how most of those amino acids are converted into alpha keto acids, and glutamate safely transports that ammonia. That ammonia gets into the urea cycle via carbonyl phosphate, and we get in the urea cycle urea and ornithine, which keeps going through the cycle. So I hope you learned something. This was a little bit longer of a lecture. This is all of chapter 18. Feel free to ask me any questions that you may have. Have a safe and blessed day.